So, good morning, everybody, and welcome to the 6th presentation of our Open Educational Resource Course Collection. I am getting ready to hear from the fabulous Anatomy and Physiology team, and I will turn this over to Dr. Brooks, who will take it away.
Thank you so much. So welcome everyone. Thank you so much for joining us and coming to share and learn about our Anatomy and Physiology 1 course content development. So I just wanted to first start out by sharing with you our landing page that we have created for you, where you can access all of the content and information related to the development process. We will talk about our process, but first I wanted to share our fabulous team. We had a team of 5 faculty experts from throughout the UNC system who teach anatomy and physiology courses, both face-to-face and online. And this team was led by our instructional design team from North Carolina Central University, myself, of course, as well as Dr. Zakaria Jwaibi. Our team librarian also supported us, Samantha Harlow from the University of North Carolina at Greensboro, as well as our fabulous system office liaison, John Fauci. So when you are in the website, once you access the website from the main system page, you will be able to explore the modules. You'll be able to access the recording of this webinar, as well as see the student learning outcomes and the various modules within the course. So when you're ready to download the content that we have created for you, just select one of these links and it will take you directly to that content collection where you will find a wealth of instructional resources, learning activities, and assessments. As you are thinking about enhancing your course, we do encourage you to consider these various best practices as you are incorporating these tools, modifying them to fit your students' needs, as well as your various instructional preferences and practices. And then we also encourage you to contact your institutional instructional design and faculty development support teams. You access this link, you will be able to go directly to your institution's Office of E-Learning, Center for Teaching Excellence, faculty development, the organizations and offices that are available to support you. Now, as far as our project development, each week we were very intentional about meeting with targeted objectives and agendas. In the very first meeting, we were able to pare down a variety of student learning outcomes for the Anatomy and Physiology 1 course based on what is commonly taught across the UNC system. And as you saw, we were able to develop the SLOs that were selected for this course. We were also able to identify potential themes for the various modules. And at the end of each meeting, we followed up with next steps related to what the team was expected to do to ensure that we met our various goals. Now, one of the immediate goals was the development of the prototype first module that laid the foundation for subsequent module development. And Dr. Jwaibi will expand on that in just a bit. But one of the key components of that was to provide an opportunity to build off of the student learning outcomes for the course and to suggest what would be the module level objectives for that prototype module. So we democratically voted on the module level objectives. The faculty experts voted on those objectives, and we were able to build them into what you will see in each module, which is the module mapping matrix that highlights not only the SLOs of the course, but the objectives for each module, as well as the various assessments, learning activities, the aligned SLOs and module level objectives, the various instructional materials, and any course technology necessary to complete that particular activity. We also utilized a transparent assignment sheet that you can also use as a guide to help you understand how to utilize the various tools, assessments, activities that you will find in the modules. And it's broken down into what are the learning objectives for each of the various learning assets, a description and purpose of each one, as well as guiding questions to help you think about what would be the purpose and description, step-by-step instructions for your learners, as well as the assessment or the evaluation criteria, and any materials and technology needed. We then prototyped our module to show what that would look like in an LMS. So here's an example in our Blackboard LMS at NCCU of what these assets may look like. Our faculty then took charge of the subsequent modules. They were responsible for developing the subsequent modules and did so by first suggesting the module level objectives for the first set of 5 modules. We worked on 5 modules for each 2-week period. And then the developers, the lead developers for those modules, selected what would be the final MLOs for that particular module. And that is how we ended up building out the 11 modules that you will find in our course collection. So Dr. Zouaibi will now expand on our prototype development process and how we were able to do so collaboratively and efficiently over this 5-week period.
Thank you, Dr. Brooks. Good morning, everyone. My name is Dr. Zakaria Zouaibi, and I am a senior instructional designer at North Carolina Central University. What I'm going to do is I'm going to highlight our design philosophy. So to expedite the creation of this course collection without jeopardizing its quality, we relied on 3 main principles. First, we adopted backward design as our course design approach. And then we decided to implement a prototype-based development process. And finally, we also use quality matters standards as our quality benchmarks. So we relied on backward design because it allowed us to design this course in a purposeful manner, and it helped us ensure alignment across all course components. By doing so, we were able to focus on quality rather than quantity. And what I mean here is that when we identified and wrote our learning objectives first, our team was able to develop appropriate assessment, was able to design meaningful learning activities, and was able to curate effective instructional materials, as well as incorporate effective instructional technologies. I also would like to add that we implemented backward design not only at the macro level, at the whole course level, but also at the micro level as well. We always started with learning objectives first to create every single learning object. So thanks to alignment, which was achieved by the implementation of backward design concept, every instructional material, every task or activity, and every assessment has an instructional function within this course collection. This means that learners will be meaningfully engaged with the content and will not perceive these learning assets as just futile, busy work. As you can see, there are 4 main stages in this prototype-based development process. In the first stage, our team identified and created student learning outcomes. As Dr. Brooks mentioned, we were able to list the potential themes or topics that would be our learning modules. Then we collaboratively created module level objectives for module 1, which was our prototype. In the second and third stages, our subject matter experts collaboratively developed a prototype. They created its assessment, selected the instructional materials, developed the learning activities, and chose the instructional technologies as well as lab component. And then the team discussed this prototype and agreed on its structure as well as the pedagogical sequencing of it. After that, the course mapping matrix was utilized, and the purpose was just to make sure that all the course components are well aligned. In the last stage, as you can see, our faculty members selected the modules they wanted to work on and started replicating the structure of the prototype module. Once all the modules were completed, we conducted a review of all the modules, and faculty made modifications as needed. Our rationale for adopting a prototype-based process was not only to expedite course development, but also to provide just-in-time training as well as practice opportunities for our subject matter experts. We thought, and we were wrong, that some faculty members might not be familiar with the online course development process and the implementation of some design concepts. However, we were lucky and fortunate to having worked with a group of faculty members who are not only experts in their fields, but pedagogically competent as well. I would like to add that the whole course development process was scaffolded to help our faculty members in this endeavor. We made sure to include templates, samples, and other resources, and those are the hyperlinked items in that document, as you can see. Throughout the whole design process, we were also engaged in one-on-one and group consultations, and we provided continuous feedback on each learning asset created by our faculty members. And I just want to emphasize here that when I said we, I meant all the team members, not just the instructional designers, but everybody was providing feedback to and supporting each other. Frankly, it was a great honor to having worked with such a fantastic team. And I think maybe we have created a learning community here, hopefully. Anyway, so another rationale for implementing prototype-based process was that by creating a prototype at an early stage of course development, we were able to identify and rectify any potential design deficiencies and learning gaps early, making the process more efficient and effective. We believe that if we did not use this prototype-based process, the likelihood of having different modules would be very high, making it a little bit more difficult or even impossible for our team to review and modify everything under such time constraints.
Thank you, Dr. Jwaibi. So now our team librarian, Samantha Harlow, is going to share the way in which we curated, identified, and documented each of the learning assets and instructional materials in the course.
So hello, my name is Sam Harlow. I'm the online learning librarian at UNC Greensboro, and I recently in the last year took over all of our OER initiatives with my colleague Melody Rude, the student success librarian. So now that you know about the design process of this course, I'm going to talk about how we kind of made this, what you're seeing on the screen, this repository of learning objects. So as these great faculty members and instructional designers were going through this design process, what I did was we pulled in the learning objects into this spreadsheet to create this course materials metadata, which the most important parts were really the citation and the license, so that when you're deciding to use this material in your anatomy and physiology courses, that you can quickly see what you're going to be citing in terms of building your own materials, connecting to your own modules, as well as the license. So whether or not you can create a derivative, whether it's a Creative Commons license, which you can do reuse and create derivatives from, a lot of them, and how you would cite them when you're creating these derivatives. So this is a sheet that is linked to that Google site that then you can quickly also see which module it's connected to, the topic, as well as what MLOs, the module learning objectives, it's meeting.
Thank you so much, Samantha. So now Dr. Sean Moore from East Carolina University is going to share a learning asset that he developed within Module 2, Histology.
Hello, everyone. I'm going to share my screen. You see? All right. So hello, everyone. As mentioned, I'm Sean Moore from ECU. I'm the director of the Center for STEM Education there and the educational advisor for the Brody School of Medicine. And so I'm going to jump right into the histology module. So you can see here that all the activities have an associated learning transparency activity sheet. And this one has a specificity for the overview of the 4 basic tissues within the human body, any associated cells, cell surface modifications, or accessory structures will be reviewed in this particular module. Now, also to supplement the students' ability to kind of acquire information, we have an OpenStax textbook chapter for their review, as well as an associated PowerPoint and an outline. And I always emphasize for students to annotate their outline to fill in any content gaps. There's also a video to watch that'll help solidify some of the concepts, and that also should be used for annotation. The culminating activity here is a histology game based on the platform of Jeopardy. And so I'm going to share that with you right now. Can you see this, hopefully?
Yes, we can.
Awesome. And so this particular activity is very versatile. You can use it as an assessment if you'd like for a score, or you can use it just as a synchronous learning activity. So it kind of gives you an opportunity to teach students how to use error analysis to bridge learning gaps. And so this can be paired with other technologies such as Kahoots, which is something like a online free clicker type of technology, or Turning Point if your institution has that. And so we can kind of go through a few samples. However, this does have an MP3 download of the Jeopardy theme song. So we'll just kind of go through one. Let's do Nervous Tissue for 2. And so this is a multiple step question. So at number 2, multiple layers of Schwann cell membranes, which are lipid rich, wrapped concentrically around number 3. Of course, this would be myelin or the myelin sheath. Another question associated with this would be at number 1, the area void of number 2. So you have to know number 1 in order to get number 2. And of course, that's the node of Ranvier. And so you can use this to kind of fill in any specific knowledge gaps. There is a return to home button right here. And so this will take you back to the home page, and it'll also shade in the question that was already asked. Let's do one more. Let's do muscle tissue for 2. Please, Alex. This striated tissue is part of an organ whose primary goal is to contract, leading to a sufficient pressure needed to transport blood, gases, nutrients, and wastes. Of course, this would be cardiac muscle. And there's another associated question, this structure here at the tip of the pointer. So with this technology, I was able to use an open source virtual microscope. And so it allowed me to customize the micrographs. And so I was able to use my digital pen to draw in any particular arrow or outline any particular structures or tissues associated with that. And then you can go back to the home, and that question will be, of course, grayed out. Now, once you've gone through this, there is an assessment. And one of the challenges has always been, how do you, in a digital world, produce a kind of a hands-on lab activity, particularly an exam, such as a lab practical. And so using Google Forms, I was able to develop a lab practical exam. And there's also a learning sheet for that as well, that'll reiterate all the learning objectives and give the purpose of this particular assessment, which is to determine how well you learn histology. And so there's a link that will take you to the Google form here. There we go. Yes, everybody can see that? Good. And so this is set up the way you would set up a station. The old school method, of course, would be to get a microscope, a box of slides, put it on the microscope, and then start developing questions. You may have to sift through a couple. And so that's the approach I took with this. So this was very painstaking. It took a lot of time. However, I was able to pinpoint exactly a good representation of a tissue. So this would be like station number 1. There are associated questions with that. Once you're finished, you click on next, and you go to the next tissue. And so with this, there's an answer key embedded. And so name the tissue presented. Of course, this is adipose tissue. The specific vacuolated cells that are predominantly seen in those tissue. This is the adipocytes. And then this is kind of interesting, one function of this tissue. So for many tissues, there's several functions. And so one would be insulation, energy production, and protection. And so with that, that takes me to the end of my 5 minutes. And should I stop sharing, Rachel? Okay.
Yes, please. Thank you so much, Dr. Moore. That was wonderful. So now we have Dr. Shankari Sumayaji from the University of North Carolina at Charlotte, who will share with us content from Module 3, the integumentary system.
Thank you, Rachel. Can you guys hear me?
Very well, yes.
Okay, thank you. So good morning, everyone. I am Dr. Shankari Somayaji from UNC Charlotte, and I have been teaching AMP 1 and 2 for a year now. Working on this project has been an amazing learning experience for me. And I am excited to share with you an activity that I have curated for one of the modules that I was assigned, which was the integumentary system. So I think one of the areas that has been the hardest to simulate in the online format for AMP has been a hands-on lab experience. So today I have chosen to share with you a virtual lab titled Investigating the Skin that I adapted from the HASPI website, which also has some other excellent AMP resources. So the specific learning goals are listed within this assessment sheet as well. So within the sheet, you can see how we have the student learning objectives, which were common throughout all the modules in this project. And we also have, I also have these MLOs, which are unique to this particular module. So as part of the activity, these are some of the learning goals that the students would accomplish once they work through this lab. So within the sheet, I have also included notes for both the student as well as faculty about how they can easily adapt this lab, both in terms of format as well as materials required for the lab. So let me jump over to the actual lab itself, and let me highlight a couple of reasons why I think this lab would fit that bill and check all those boxes for an online course. Okay, so as part of their first activity, students would visualize evaporative cooling as a homeostatic mechanism of the integumentary system. So if you see here, using very simple materials at home, they can work with a partner and they can easily be able to do this activity from at their homes. The next thing they would do as part of their activity too is they would again map out different kinds of tactile receptors like nociceptors, mechanoreceptors, thermoreceptors. They would be able to do all of those mapping of those receptors, again using very simple materials such as horsehair, steel pins, cup of ice, etc., all things which are easily accessible. So as you can see, the students get a hands-on interactive experience where they can actively engage with the material for a relatively large pedagogical payoff. The other neat thing that I really like about this lab is that it's easily accessible, affordable, and also flexible. So if students do not have horsehair at home, they can easily use a paintbrush, or they can use a feather. Or if they don't have rubbing alcohol for the first activity, they can substitute it with a nail polish remover. It's also a highly adaptable lab. So in the sense that if faculty members are teaching their labs completely online, then students do get a very hands-on experience working on this with a volunteer at home. And the other nice thing is it doesn't have to be a biology student who they are working with. In fact, they can work together with their younger siblings who might be in middle school or high school, and they can all engage in these interactive activities and kind of keep each other busy as well. Instructors can also assign this if they are using a hybrid format. So I know, for example, UNCC, in fall, we are having our students attend the AMP labs in a hybrid setting. So some of them will come into the lab for the first half an hour and then go back and work on online assignments, and the other half will then come in and do the same. So this can easily be assigned as a virtual lab for students who are working at their home or from their dorms, and they can then come into the lab to work on activities that would actually mandate those wet lab resources, which would be available only within a lab environment. So in the last part of this lab, they would determine the sensitivity of skin using very accessible materials like a compass and a ruler. But the nice thing about this particular activity is they also get to plot data, they get to analyze the data, and then they get to infer the physiological significance of the data. So for example, in this activity that they collect as part of their 2-point touch activity, they would notice that the receptor density is the highest or should be the highest in the lips and on the fingertips. And that will help them make a correlation about the importance of these areas in gathering this sensory information. So I like how also they're able to engage and develop those higher order critical thinking skills by plotting data, evaluating their evidence, and drawing conclusions. So that is what I have to share with you today. Thank you.
Thank you so much, Dr. Sumayaji. So now we have Dr. Marta Kleesoth from North Carolina State University, who is going to share a learning asset from Module 4, the skeletal system.
Sorry, can you hear me? Okay, excellent. So I wanted to reiterate how exciting this project was to be able to share and engage with such a wonderful team. And it really led to some creative ideas, I think. And so one of the things that I wanted to show would be this learning portfolio that came out of some of the discussions and ideas that we had. So this learning portfolio, like all of our activities, was based on this transparent learning asset or activity that we have. So just as was shown before, I've gone through and I've taken this learning guideline here and I've aligned it with the student learning or the student learning objectives or outcomes, and then the module learning objectives, so that it's tied together so the student can clearly see how this is effective. When you look at this, I've outlined the step-by-step instructions as has been previously shown. And one of the things I wanted to point out is that we've provided links in all of our assessments to look at different assets that we found that will help them. But really, this is about the pedagogical aspect of what's happening here. When we're looking at this learning portfolio, the idea is that we want to go ahead and engage the students with two main goals. One, they're taking notes, and that's a really important point because especially in an online format, when students are referencing online content, in some cases they're unsure how to utilize those content and they may spend a lot of time on it with very little learning gains. So there was instruction and links on different ways to approach this and different ways to find note-taking skills that would work for you. They will also create an educational visual component, and this is their opportunity to merge together this learning. As far as the learning portfolio, one of the main reasons to go ahead and approach it in this way is that the students now are not only creating but sharing this digital content. One of the things that you're going to be able to see through this process is that, I'm sorry, just one second. One thing you'll be able to see through this process is that they are creating and making meaning of the content that they're giving, which is extremely important in an anatomy and physiology course. Also, it's designed to build the students' skills. One of the things that they'll do is they'll be using the skills that they have approached in this learning portfolio throughout their academic career. And we've provided instructional support and guidance so that the instructor now has video tutorials that can also be shared with the students on how to create this digital media, and then also the pedagogical guidance. So different ways that this can be engaged. And this is just showing you some video support that we have provided for this. One of the big advantages to this process is that it offers a lot of flexibility, and it offers the ability for the students to collaborate. It gives you collaborative experiences, and it can allow you to not only provide written components to expand upon, but have a record of those, and you can see growth throughout that entire unit and or through the course. And that's part of that content scaffolding that you'll see, and the fact that if done properly, this can promote metacognitive skills. So I want to show you the learning portfolio. The instructors are provided with a template that they can share with their students. And when you open the homepage of this, it does align immediately with the module learning objectives, and it lets them know what parts and components that they're responsible for. So I have a sample one filled out here for you. This is an example of the learning component that they would have. And they have a variety of different things that they can do. They can go ahead and do concept maps. They can create infographics. They can do videos. They can do models and take pictures of them. The point is that they are going to go ahead and create something that integrates the information that they have collected and then provide a description of what that learning value is and why it's important. And you can see they will provide their notes here. So the note tissues, for example, they don't have to be, really digital savvy or tech savvy to do this. They can just take pictures of their handwritten notes and upload them into this area. It gives them an area to collect this information so it can be reviewed. And also then it allows the instructor to review it and see where things went wrong. The last thing I wanted to mention is you can expand upon this so that we have, in this case, a lab component. This will allow the students now to upload this lab content so they can tie the lab materials with it. You can also have a page that becomes the assessments. So now you can directly tie that content with their notes to the activities that they've done and recorded, and now you can tie that to their assessments and really be able to walk through and see how they've integrated all of this information.
Thank you so much, Dr. Kleesoff. That was phenomenal. So we now have Professor Tawana LeBennett from North Carolina Central University, who is going to share content developed in Module 7, which is on organization of nervous tissue.
Thanks, Rachel. Hi, everybody. I'm Tawana LeBennett, faculty in the Department of Biological and Biomedical Sciences at North Carolina Central University, and I teach anatomy and physiology. And I chose the neurophysiology virtual lab to demonstrate in this course, to show its versatility and in highlighting neurons and nerve actions. First, we start with the transparent activity sheet, a sheet designed to provide a guide that informs students of the purpose of the exercise, and it defines the precise practice activities that the student should perform. As indicated in this transparent activity sheet, students will navigate over to the HHMI website, where they are instructed to utilize the tabs, and they will follow the procedures beginning with the leech dissection. And as you can see, this leech dissection allows the students to see structures, internal organization of the nervous tissue, and this is noted just underneath the skin of the leech. During this dissection process, electrodes were attached to measure the electrical activity of the neurons. They were later injected with a fluorescent dye to enhance the observations. Using various instruments, nerves were stimulated to elicit a response or an action potential. This created an atlas for students to reference these catalog cells, which were available again for the students to review as they then move forward to the probe and identity phase. In the probe and identity phase, students began to review and determine which cell they are going to select. So from these selected cells, they will use their instruments, either a probe, a brush, or a forceps, to begin reviewing how nerve activity occurs within this organism. As can be seen, also students can access information in the drop-down menu. The drop-down menu can refresh their memory on such terms as an action potential, a resting membrane potential, and then using their identification and inspection sheet, which we'll show you, students document their observations by doing some sketches, some notes, and then they will use their sketches of the individual nerve cells that they've been able to locate and select to compare to the slideshow that's presented here. So these are histological slides showing a series of nervous tissue slides, which can be compared to the slides that to the sketches that they have already done in their comparison. Now, with these slides, again, they can observe a variety of different cells. And this series of slides also displays some of the nervous tissue, cerebellum, the cortex, the ganglia. Now, using these slides, cell tissues, then students can compare the slides that they were looking at previously to the ones that they observed in the leach dissection. These identified leach cells, then they compare their structure, look at the similarities, look at the differences, and then they will return from here to the transparent activity sheet, back to the transparent activity sheet, where they will be instructed to conclude the activity with a set of summary questions on their observations and experiences. And this was a great opportunity for me, and I enjoyed every step of the way. Thank you so much.
Thank you so much, Professor LeBennett. So to conclude our sharing today of the content in our collection, we have Dr. Jason Wingard from the University of North Carolina at Asheville, who will be sharing content developed in Module 9 related to the peripheral nervous system.
Thank you, Dr. Brooks, and hello, everyone. I would like to start with an overview of Module 9 on the peripheral nervous system by showing you the mapping matrix for this module. It begins with links to an OER chapter on the peripheral nervous system. It has lecture slides that correspond with the OpenStax textbook and introductory videos on the peripheral nervous system. The peripheral nervous system has two major divisions, the somatic nervous system and the autonomic nervous system, and this module is divided accordingly. There are videos on different aspects of the somatic nervous system, including somatosensory function, motor function, proprioception, kinesthesia, etc. And then similarly, there are links to videos that describe the autonomic nervous system and its component parts, including the sympathetic nervous system. And there is also a case study on the sympathetic nervous system. And this module also includes practice quizzes so that the student can practice what they're learning before they get to an assessment. This module also has two virtual labs. The first one is on cranial nerves, and a very good lab to reinforce what the student is learning about the cranial nerves. And then there's a second virtual lab on the somatic nervous system. And I would like to focus our attention on this second virtual lab. So again, this lab covers the peripheral nervous system and focuses on reflex testing and also testing dermatomes and the clinical applications of both of these types of tests. So there's introductory information, and we get down to the step-by-step directions. First, in part one on the reflex lab, there is an introductory video on assessing reflexes. And then there is a lab that we are asking the students to complete. And again, there's introductory information about the components of a reflex. This reinforces information that they will have just seen in the video, describing the sensory and interneuron and motor components of reflexes. And then it gets into the procedures. Part A has the student testing the stretch reflex, or what some people refer to as the patellar tendon reflex or the knee jerk reflex. Goes through a bit of the anatomy and physiology of this reflex, and then it instructs the student on how to assess this reflex. And then there is a good experimental portion where it's having the student test this reflex after their volunteer, while their volunteer is reading a book. So this acts as a distraction and asks the student to assess the quality of the reflex while their volunteer is distracted. And then another experiment where the student is testing this reflex after the volunteer has done some exercise, and so therefore the quadriceps muscle is fatigued. So there are some fun interactive reflex testing. And then it goes on to instruct the student on how to do Achilles reflex testing, the biceps tendon reflex testing, and triceps tendon reflex testing. And so a big part of an anatomy and physiology lab is experiencing that hands-on, sometimes it's awkward, but it's important to work through and practice this hands-on assessment. And so this lab does a good job of doing that. And just like was mentioned before on the cutaneous labs, the student can really do these labs on anyone. And if they don't have anyone nearby to do these labs on, they can actually do this reflex testing on their own body. Doesn't work quite as well, but it's possible. There are other reflexes that are available in this lab if the instructor wants to use them. Cutaneous reflex on the patellar surface of the foot, cranial reflex, looking at pupillary reflex, and then reaction time, which is an excellent assessment of sensory and motor speed and integration. This lab ends with assessment questions on what they've just seen and done. Then part two, I'll go through quickly, is on dermatomes, and it begins with a scientific article that describes recent revisions to one of the common dermatome tests. And so a dermatome is the area of skin that is innervated by a specific nerve coming off of the spinal cord. We call those spinal nerves. And so for example, the umbilicus or the belly button is innervated by the spinal nerve coming off of thoracic vertebrae number 10, T10. And so that would look something like this. Although this picture doesn't show T10, it's showing spinal nerves coming off of the spinal cord. So this paper gives them excellent background. There's a link to a YouTube video watching a clinician do dermatome testing. And there's an interactive practice quiz where the student can, see if I can access this quickly, choose their view and select on an area of skin to find out what dermatome level that is. This is just a way to practice. So again, that's T10, innervating the area of skin around the umbilicus. And then there is a link to a lab. And this lab uses the ASIA scale, which is the International Standards for Neurological Classification of Spinal Cord Injury, which is a very commonly used test internationally for assessing spinal cord injury and level of spinal cord injury. So the student would be asked to complete this test on a volunteer. And basically it has them testing the different dermatome levels as shown here with two objects, a sharp stimulus, like the end of a paper clip would be fine, and a light touch stimulus, like a cotton ball, and going down the dermatomes and assessing the function. And then in case they're not assessing a person with a spinal cord injury and they'd be interested in seeing how that's done, this module includes a second scientific paper that shows an example of this testing done on a person with spinal cord injury. And so we see here normal sensation through cervical vertebrae number 7, and then the absence of sensation below that level. And so this gives a nice, actually 2 good clinical examples of using this type of test. And then this module ends with a link to the assessment for the somatic nervous system, which should be completed after they've run through these labs. Thank you.
Thank you so much, Dr. Wingert. So again, we are delighted to have been able to create these assets for you all over the course of these past 5 weeks. And we encourage you to access the course collection via the link that was shared in the chat by John Fauci. And once again, you can access the modules by clicking on Explore the Modules, and you will have direct access to all of the learning assets that were created directly in their folders, as well as the guide sheet that explains how to utilize each one. And if you need any assistance, again, you may contact your individual institutional, faculty development, and instructional design support teams by clicking on the logos to get assistance in applying these various learning assets to your LMS. Thank you so much, and we're so glad to have been able to share this with you.
Thanks, Rachel. That was fabulous. I learned a lot, got a little bit grossed out. Cool. Are there any questions from the group?
Hi, am I coming through?
Yes, you are.
Hi, this is Bob Pogue. I'm down at University of North Carolina, Pembroke. I'm not teaching a section of AMP1 this semester. I'm teaching AMP2, and it looks like it breaks down very similarly to the way you have prepared and presented it. Are there plans to expand this module to include the other body systems that are not included that were not included today?
That is a great question. So when you take a look at the assets, you'll see that there are a number of other body systems that are present. I will let Michelle go ahead and share about murmurings, I don't know. But just to provide a little context, the reason why the systems that were selected for this particular course collection or the way in which it was done was just kind of scanning and canvassing which are the common ones that would often be taught and learners would be expected to have successfully mastered upon completion of Anatomy and Physiology 1. There were discussions regarding certain systems often being taught on different campuses in AMP1 where others may be taught in AMP2. So there were discussions about that, and we have suggestions and ideas about moving forward. But this was designed, and you'll find inside of the different the learning guides that you'll find in the folders, there are instructor notes that provide support and guidance as it relates to how you might tie this in with what you will be covering in AMP2, or if this is more appropriate to combine with other systems while you're teaching AMP1. So this was not designed as an all-in-one course to be taught as is. It was intentionally constructed such that alignment is present and it can be used together, but please understand that you can use these modules individually. We were trying to be very explicit so that you can see how it aligns and may support alignment with your course level objectives and module objectives, or you can pull out individual learning assets. They're combined in folders with the guide to go along with them. So however it best suits you, please feel free to manipulate them and go forth.
Well, I would have loved to have had this in March when I was teaching. I mean, you covered the same material that I covered in the spring. And now I just happen to have a different section. But so it sounds like expanding things to include respiratory, digestive, circulatory, all that. That's I shouldn't expect those to magically turn up between now and August. Is that a safe statement?
That would be a safe statement. We have ideas and would love to see this expand further to include, we did when we were thinking about the types of systems, what systems to include in that original list, those were listed there, and then we had to prioritize based on one, the time that we had and the amount of content we would be able to develop, but also what are the most common ones and how could this be most useful and flexible across the system. And so we ended up with the modules that you see here, those 11.
Great. I will share it with my colleagues who have who have 2110. It looks like a great resource. Thank you.
Thank you.
Wanted to ask everybody to please fill out the survey that we put the link in the chat a couple of times. We would just appreciate your input. And Rachel and team, thank you very much. You guys did a fantastic job. I'm giving you a round of applause. Yay. Well done. Happy dance.
They make us look good. We had a phenomenal team. Absolutely wonderful.
Yes, our UNC system faculty absolutely rock and have outdone themselves on this project, no doubt. As have our instructional designers and our librarians.
That's
and our liaison. Yes.
Yes. Yeah. So yeah, this is Sean Moore from EC. I really do just want to thank the instructional design team. I know what it's like not to have a lot of support and resources. And, you know, Rachel, Zach, and Sam, you guys are invaluable. And I'm grateful that you were able to kind of guide us through this process. You know, I think your group as a team really made all of this digestible in the 30 days that we had to complete it. We all went through some long nights. You know, Rachel got me straight several times on what I needed to do and how I needed to do it. And I'm grateful and thankful for all your support, as well as my teammates. Collaborating with you guys has been awesome. But I did want to get that on the recording and on record for public consumption, because it's important and it's critical to the success of faculty as instructors. So, you know, kudos to you. Central is very lucky to have you all.
Thank you so much.
Thank you, Sean.
Okay, luckily I got that on recording, and I'm going to stop the recording.